Tonight's lecture is over Chapter 6, DNA and Gene Expression. And for this chapter, our learning objectives are to be able to describe what DNA is and what it does. We should be able to explain the process of gene expression involving transcription and translation. And then we should also be able to explain the causes and effects of damage to the genetic code. On a rainy night in November 1998, two young women were attacked in Baltimore. One was able to escape, but the other was stabbed to death. Although it was dark, and she was able to see the attacker for only a few seconds, the surviving victim was able to give the police a description that enabled them to produce a composite sketch of the suspect. A week later, a friend of the victims contacted the police saying that he recognized the man in the sketch. He identified 24-year-old Malcolm Bryant, and in a photographic lineup, the surviving victim identified Bryant as the man who attacked her. Based on the eyewitness identification, Bryant was arrested and charged with first-degree murder. Although multiple witnesses testified that Bryant was in a nightclub at the time of the crime, a jury convicted him and sentenced him to life in prison. Bryant maintained his innocence, but his appeals were all rejected. Then, in 2011 and again in 2015, attorneys from the Innocence Project successfully petitioned to get DNA testing done on evidence that had been collected at the time of the crime. Ultimately, forensic scientists were able to obtain a full DNA profile from the T-shirt of the murder victim. This profile matched a partial profile from the skin under the fingernails of the victim. It also did not match Malcolm Bryant's DNA, providing strong evidence that he was not the murderer. His wrongful conviction was dismissed, and in 2016, after more than 16 years in prison, Malcolm Bryant was released. Malcolm Bryant is one of 367 unjustly imprisoned people in the United States as of late 2019 who have been freed from prison as a result of DNA analysis. They spent an average of 14 years behind bars. 80% had been convicted of sexual assault, 36% had been convicted of murder. In 70% of the cases, inaccurate eyewitness testimony played an important role in the guilty verdict. And recall this from Chapter 1. We talked about how experiments revealed the unreliability of eyewitness identification. In this chapter, we take a close look at DNA, the molecule responsible for Malcolm Bryant's exoneration and the deferred justice served to 366 other people. All living organisms, people, plants, animals, bacteria, and otherwise, carry DNA in almost every cell of their bodies, with just a few exceptions. Like a social security number, every person's DNA is unique. The only exception to this is identical twins. In addition to being contained in our living cells, our DNA exists in what we leave behind. It is present in our saliva, hair, blood, and even the dead skin cells that fall from our bodies. It's nearly impossible to read or listen to a news report these days without being informed that yet another complex human characteristic or trait has been linked to a newly discovered gene. Keep in mind, however, that such reports often oversimplify the complex connections between our genes and these traits while downplaying the myriad of other factors that significantly influence the traits. We'll explore these issues and more, beginning with a look at the structure of DNA and how it contains the information for producing organisms. And that brings us to our take-home message for section 6.1. DNA is a molecule carried by all living organisms in almost every cell of their bodies. DNA contains instructions for the functions of every cell. Because every person's DNA is unique, and because we leave a trail of DNA behind us as we go about our lives, and even after, DNA can serve as an individual identifier. Moving on. Beginning in the 1900s and continuing through the early 1950s, a series of experiments revealed two important features of DNA. First, molecules of DNA are passed down from parent to offspring. Second, the instructions for how to create a body and control its growth, development, and behavior are encoded in the DNA molecule. Given that DNA must hold the instructions for how to produce every possible type of structure in every living organism, scientists in the 20th century raced to determine the chemical structure of DNA and to understand how the molecule is assembled and shaped so that it can hold and transmit so much information. Many formidable scientists rose to the challenge. When the American Linus Pauling began investigating the structure of DNA, he had already won a Nobel Prize in chemistry for his work on elucidating the structures of molecules. Simultaneously, in England, Maurice Wilkins and Rosalind Franklin were devoting their research to the task and produced X-ray images of DNA that were critical to decoding its shape. But it was Englishman Francis Crick and an American James Watson working in Cambridge, England, who happened to put all the pieces together to figure out the exact structure of DNA. Moving on, DNA, also known as deoxyribonucleic acid, is a nucleic acid, a macromolecule that stores information. It consists of individual units called nucleotides, which have three components: a molecule of sugar, a phosphate group containing 4 oxygen atoms bound to a phosphorus atom, and a nitrogen-containing molecule called a base. The physical structure of DNA is frequently described as a double helix. You can visualize the double helix as a long ladder twisted around like a spiral staircase. And we see that image on the left-hand side of our slide, and this image is also Figure 6.4 on page 152 of your book. The DNA molecule has 2 distinct strands, like the vertical sides of a ladder. These are the quote-unquote backbones of the molecule, and each is made up of 2 alternating molecules: a sugar, then a phosphate group, then a sugar, then a phosphate group, and so on. The sugar is always deoxyribose, and the phosphate group is always the same too. It is the shapes of the molecules in the backbone that cause the DNA ladder to twist. The alternating sugars and phosphates hold everything in place, but the rungs of the ladder are where things get interesting. Attached to each sugar and protruding inward like half of a rung of the ladder is one of the 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. When discussing DNA, these bases are usually referred to by their first letter, A, T, C, G. Both backbones of the ladder have a base protruding from each sugar. The base on one side of the ladder, one strand of the DNA, binds to a base on the other side via hydrogen bonds, and together these base pairs form the rungs of the ladder. They pair up predictably. Every time a C sticks out from one side, it forms hydrogen bonds with a G on the other side, and vice versa. In other words, G always bonds to C, as C always bonds to G. Similarly, each T forms hydrogen bonds with an A on the other side, and vice versa. Get it? A always bonds with T, and T always bonds with A. Because of the base pairing rules, if we know the base sequence of one of the strands in a DNA molecule, we can figure out the complementary sequence in the other. This is why a DNA sequence is described by writing the sequence of bases only in one of the strands. If a human DNA molecule were really a twisted ladder, it would be a very, very long one. One molecule of DNA can have as many as 200 million base pairs or rungs. How does such a molecule fit into a cell? The rungs are small, and the twisting of the molecule, twists upon twists, shortens it considerably. Think about how, if you repeatedly twist your shoelace around and around, it becomes shorter and shorter. That's what happens with DNA. And that brings us to our take-home message for section 6.2. DNA is a nucleic acid, a macromolecule that stores information. It consists of individual units called nucleotides, which comprise a sugar, a phosphate group, and a nitrogenous base. The sequence of bases on one side of the ladder-like molecule, one strand of the DNA, complements the bases on the other side, or the complementary strand. Moving on, one of DNA's most amazing features is that it is a common language used to code for structures in almost every single living organism on Earth. Thus, DNA is like a universal language whose letters consist of the bases A, T, C, and G. Note that the sugar-phosphate backbone serves only to hold the bases in sequence, like the binding of a book. It does not convey genetic information. Another analogy may help you to understand the information-containing aspect of DNA. You can think of the organism's DNA as a cookbook. Just as a cookbook contains detailed instructions on how to make a variety of foods, an organism's DNA carries the detailed instructions to build the organism and to keep it running. And just as a book can be viewed as a sequence of letters, with the book's meaning determined by which letters are strung together and in what order, a molecule of DNA can be viewed as a sequence of bases. Letters don't have much meaning on their own, of course, but when they are put together into words and sentences, their order holds a great deal of information. Similarly, the sequence in which bases appear in a molecule of an organism's DNA makes up a code that specifies the detailed instructions for building polypeptides. Upon processing and folding, these polypeptides become the numerous and varied protein molecules that make up an organism, whether it is a one-celled amoeba, a giant oak tree, or a biology student. Moving on, in prokaryotes, including all bacteria, the building instructions are contained within circular pieces of DNA. In eukaryotes, including human, this information is laid out in long linear strands of DNA called chromosomes in the nucleus. Rather than being one super long DNA strand, eukaryotic DNA exists as many smaller, more manageable pieces. Humans, for example, have 3 billion base pairs divided into 23 unique pieces of DNA. Because we have 2 copies of each chromosome, one from our mother and one from our father, we have a total of 46 chromosomes in each cell. The full set of DNA present in an individual organism is called its genome. Within the long sequences of bases in a cell's DNA molecules are relatively short sequences, on an average about 3,000 bases long, called genes. You may hear the word gene misused in the media, as if a gene were a mysterious force controlling our bodies and behavior. It is not. Here we'll be precise. A gene is a sequence of bases, or more precisely base pairs, in a DNA molecule that carries the information necessary for producing a functional end product, usually a polypeptide or an RNA molecule. Nothing more, nothing less. The location or position of a gene on a chromosome is called its locus. Moving on, each gene is the instruction set for producing one particular molecule, usually a protein. For example, a gene in silk moths codes for fibrin, the chief component of silk. As another example, a gene in humans codes for triglyceride lipase, an enzyme that breaks down dietary fat. Within a species, individuals sometimes have slightly different instruction sets for a given protein, and these instructions can result in different versions of the same characteristic. These alternative versions of a gene that code for the same feature are called alleles, and they function like alternative recipes for chocolate chip cookies. Any single characteristic or feature of an organism is referred to as a trait. For example, the color of a daisy's petals is a trait. The instructions for producing this trait are found in a gene that controls petal color. This gene may have many different alleles within a population. One allele may specify the trait of orange petals, another may specify yellow petals, and yet another may specify purple petals. Similarly, one allele for eye color in fruit flies may carry the instructions for producing red eyes, while another, slightly different allele may have instructions for producing brown eyes. Ultimately, though, the trait may be identified not just by the genes an individual carries, but also by the way those genes interact with the environment. And that brings us to our take-home message for section 6.3. DNA is a universal language that provides the instructions for building all the structures in all living organisms. The full set of DNA that an organism carries is called its genome. A gene is a sequence of bases in a DNA molecule that carries the information necessary for producing a functional product, usually a polypeptide or an RNA molecule. Moving on, it is debatable whether humans are the most complex species on the planet, but surely we must be more complex than an onion. Comparing the amount of DNA present in various species in terms of both number of chromosomes and numbers of base pairs, however, reveals a paradox. There does not seem to be any relationship between the size of an organism's genome and the organism's complexity. The description earlier in this chapter about what DNA is and how genes code for proteins is logical and tidy, but it doesn't completely explain what we observe in cells. In humans, for example, genes make up about only 1% of the DNA. However, in many species, the proportion of DNA that consists of genes is even smaller. In almost all eukaryotic species, the amount of DNA present far exceeds the amount necessary to code for all the proteins in the organism. Moving on, the fact is, a huge proportion of the base sequences in DNA do not code for proteins, and most of these have no known purpose. When it comes to what was first observed, some biologists even referred to this non-coding DNA as quote-unquote junk DNA, although as we'll discuss later, some of the non-coding DNA does have other functions. Moving on, bacteria and viruses tend to have very little non-coding DNA. Genes make up about 90% or more of their DNA. It is in the eukaryotes, though, with the exception of the yeasts, that we see an explosion in the amount of non-coding DNA. Non-coding regions of DNA often take the form of sequences that are repeated sometimes thousands or even hundreds of thousands of times. Often, these repeats exist because some DNA sequences can make copies of themselves, and the copies can move about throughout the genome. Occasionally, the non-coding DNA consists of gene fragments, duplicate versions of genes, and pseudogenes, or sequences that evolved from actual genes but accumulated mutations that made them lose their protein coding ability. About 25% of the non-coding regions occur within genes, in which case they are called introns, in contrast to the coding regions, which are called exons. The remaining 75% of the non-coding regions occur between genes. The role of this non-coding DNA is not completely understood. Recent evidence reveals that some of the non-coding DNA does, in fact, encode extremely short RNA molecules, approximately 20 nucleotides long, that function in gene regulation. They act as a switch that regulates when genes are turned on or off, or as a volume knob that influences the amount of the gene products produced. Non-coding DNA may also serve as a reservoir of potentially useful sequences. In any case, the label junk DNA is not necessarily an accurate description of all of the non-coding DNA. This brings us to our take-home message for section 6.4. Only a small fraction of the DNA in eukaryotic species is in genes that code for proteins. The function of much of the rest is still poorly understood, but at least some of it plays important roles in the cell, such as gene regulation. Moving on, having a recipe for chocolate chip cookies is not the same thing as having the actual cookies. Similarly, having a wealth of hereditary information about how to build muscle cells or leaf cells is not sufficient to produce an organism. Think about it. An organism's every cell contains all of the information needed to manufacture every protein in its body. This means that the skin cells in your arm contain the genes for producing proteins found only in liver cells or red blood cells or muscle tissue, but they don't produce those proteins. Having the instructions is not the same as having the products. The genes and strands of DNA are a storehouse of information, an instruction book, but they are only part of the process by which an organism is built. If the genes that an organism carries for a particular trait, its genotype, are like a recipe in a cookbook, the physical manifestation of the instructions, the organism's phenotype, is the cookie or any of the other foods described by the recipes. And just as you have to assemble the ingredients, mix them, and then bake them to get the cookie, there are multiple steps in the production of the molecules, tissues, and even behaviors that make up a phenotype. So how does a gene or a sequence of bases in a section of DNA affect a flower's color or the shape of a person's nose or the texture of a dog's fur, which would be a phenotype? The process involves two main steps: transcription, which is a copy of the gene's base sequence made, and translation, which is a copy that is used to direct the production of a polypeptide, which then, in response to a variety of factors, including the cellular environment, folds into a functional protein. And if we look at page 157, figure 6.11, we see this cartoon. This cartoon presents an overview of the processes of transcription and translation. In transcription, which in eukaryotes occurs in the nucleus, the gene's base sequence or code is copied into a go-between molecule called messenger RNA, or we sometimes call that mRNA. And remember that prokaryotes do not have a nucleus, so transcription for those types of organisms occurs in the cytoplasm. Transcription is like copying the information for the chocolate chip cookie recipe out of the cookbook onto an index card. The mRNA then moves out of the nucleus and into the cytoplasm, where translation allows the messages encoded in the mRNA to be used to build proteins. And this cartoon that we see on our slide is sometimes referred to as the central dogma of molecular biology. And that brings us to our take-home message for 6.5. The genes and strands of DNA are a storehouse of information. The process by which this information is used to build an organism involves two main steps: transcription, in which a copy of the gene's base sequence is made, and translation, in which that copy is used to direct the production of a polypeptide or direct protein synthesis. Moving on, if DNA is like a cookbook filled with recipes, transcription and translation are like cooking. In cooking, you use information about how to make chocolate chip cookies to produce the actual cookies. In an organism, the information about putting together proteins is used to build the proteins that an organism needs to function. In this section, we will examine transcription, the first step in the two-step process by which DNA regulates a cell's activity and its synthesis of proteins. Transcription is like copying a single recipe from the cookbook onto an index card. In the four-step process of transcription, a copy is made of one specific gene within the DNA. So moving on, the first step is to recognize and bind. To start the transcription process, the enzyme RNA polymerase, or RNAP, recognizes a promoter site. a base sequence in a gene that indicates the start of the gene and in effect tells the RNAP, start here. RNAP binds to the DNA molecule at the promoter site and unwinds a portion of the molecule as the RNA polymerase moves along the strand of DNA so that only one strand can be read. Throughout transcription, DNA is unwound ahead of the RNAP so that the single strand of DNA can be read, and it is rewound after the polymerase passes. Step 2, transcribe. As the DNA strand is processed through the RNAP builds a copy called a transcript of the gene from the DNA molecule, just as a court reporter transcribes and creates a record of everything that is said in a courtroom. This copy is called messenger RNA or mRNA because once it is created, it can move elsewhere in the cell and its message can be translated into a protein. The mRNA transcript, which is constructed from 4 different nucleotides, which have a structure similar to that of DNA, but with a different kind of sugar, ribose, rather than deoxyribose. Each nucleotide pairs up with an exposed base on the unwound and separated DNA strand following these rules. If the DNA strand has a thymine or a T, an adenine is added to the mRNA. If the DNA strand has an A or an adenine, a uracil is added to the mRNA. If the DNA strand has a guanine or a G, a cytosine or C is added to the mRNA, and if the DNA strand has a cytosine, C, a guanine, G, is added to the mRNA. Notice that the mRNA molecule contains the base uracil instead of thymine, so therefore, where the DNA alphabet is CGAT, the RNA alphabet is CGAU. And that brings us to step 3, termination. When the RNAP encounters a sequence of bases on the DNA at the end of a gene called the termination sequence, it stops creating the transcript and detaches from the DNA molecule. After termination, the mRNA molecule is released as a free-floating single-strand copy of the gene. And that brings us to step 4, which is cap and edit. In prokaryotic cells, once an mRNA transcript is produced, it begins to separate from the DNA. It is ready to be translated into a protein. It doesn't have a nuclear membrane to cross. However, in eukaryotes, mRNAs undergo extra processing before they can be translated into a protein. First, a cap and a tail may be added at the beginning and the end of the transcript. Like the front and back covers of a book, these serve to protect the mRNA from damage and help the protein-making machinery recognize the mRNA. Second, because as we saw earlier in this chapter, some non-coding bits of DNA may be transcribed into mRNA, those sections, the introns, are snipped out and the exons are joined. This is called splicing. Once the mRNA transcript has been edited, it is ready to leave the nucleus for the cytoplasm, where it will be translated into a polypeptide that will fold into a protein. And that brings us to our take-home message for section 6.6. In transcription, which occurs in the nucleus in eukaryotic cells, a single copy of one specific gene in the DNA is made in the form of a molecule of mRNA. When the mRNA copy of a gene is completed and processed, it moves out of the nucleus and into the cytoplasm, where it can be translated into a polypeptide. Moving on, once the mRNA molecule has moved out of the cell's nucleus and into the cytoplasm, the translation process begins. In translation, the information carried by the mRNA is read, and its message can be translated into a protein. Think of it this way. To read an encrypted message, you need to know the code. That is, you need to know how to translate each of the encrypted characters into a readable character. For mRNA, another type of RNA molecule holds the code. This molecule, called a transfer RNA or a tRNA, interprets the mRNA code, translating the language of DNA, coded in the linear sequence of bases, into the language of proteins, coded in the linear sequence of amino acids. Picture a molecule with two distinct ends. And you can see that here in this figure on our slide. One end of the tRNA molecule is an attachment site called an anticodon, consisting of a 3-base sequence that matches up with the 3-base sequence on the mRNA transcript. This matchup enables a tRNA molecule to attach to the mRNA molecule. Attached to the other end of the tRNA molecule is the corresponding amino acid. Moving on, each 3-base sequences in mRNA, called a codon, matches with a tRNA molecule that carries a particular amino acid. The codon ACG, for example, is recognized by the tRNA molecule that carries the amino acid threonine. Similarly, the codon CAG is recognized by the tRNA molecule that carries glutamine. For every possible codon, one type of tRNA molecule will recognize and bind to the mRNA molecule at that point, and it always carries the same amino acid. So the codon table that we see on our slide here, this is on page 161 of your book, figure 6.14, describes which tRNA and therefore which amino acid is specified by each codon in the mRNA. With 4 possible bases in each of the 3 positions of codon, 64 different codons are possible. 61 of these codons specify for amino acids, and 3 are stop sequences indicating the end of translation. Because there are only 20 amino acids, many amino acids are specified by multiple codons. UGU and UGC, for example, both specify cysteine. With just a few small exceptions, the genetic code is the same in every organism on Earth. For translation to occur, several ingredients must be present in the cytoplasm. First, there must be a large supply of free amino acids. Recall from Chapter 3 that amino acids are the raw materials for building proteins and an essential component of our diet. Second, there must be ribosomal subunits, which are the components of ribosomes, the protein production factories in which amino acids are linked together in the proper order to produce the protein. The translation of an mRNA molecule into a long sequence of amino acids, which will then fold into the complex three-dimensional shape of a protein, occurs in 3 steps. Moving on. Step 1. Recognize and initiate protein building. Translation begins in the cell cytoplasm when the subunits of a ribosome recognize and assemble around the codon on the mRNA transcript called the start sequence. This start sequence is always the codon AUG. As the ribosomal subunits assemble themselves into a whole ribosome, the attachment site of a particular tRNA molecule also recognizes the start sequence on the mRNA and binds to it. This initiator tRNA carries the amino acid methionine. Thus, methionine is always the first amino acid in any protein that is produced. And typically in eukaryotes, this initial methionine is edited out in a later protein building process. And that brings us to step 2, elongation. After the mRNA start sequence, AUG, the next 3 bases of the mRNA specify which tRNA molecule should bind to the mRNA. If the next 3 bases on the mRNA transcript are GUU, for example, a tRNA molecule that recognizes this sequence and carries its particular amino acid valine attaches to the mRNA at that point. The ribosome then facilitates the connection of this second amino acid to the first. After the first amino acid carried by its tRNA molecule is attached to the second amino acid, the first amino acid's tRNA molecule detaches from the mRNA and floats away. As this process continues, the amino acid chain grows. The next 3 bases on the mRNA specify the next amino acid to be added to the first 2 amino acids. The 3 bases after that specify the 4th amino acid, and so on. This process of progressively linking together the amino acids specified by an mRNA strand is called protein synthesis, because all proteins are chains of amino acids arranged like beads on a string. Precision in the process of protein synthesis is essential. If the tRNAs were to misread, skip, or double read any bases, they could alter the amino acid sequence and possibly the normal functioning of the protein specified by the mRNA. And moving on to the next slide, that brings us to step 3, termination. Eventually, the ribosome arrives at the codon on the mRNA that signals the end of translation. Once the ribosome encounters the stop sequence, the assembly of the amino acid chain is complete, and the amino acid chain and mRNA molecule are released from the ribosome. As it is being produced, the amino acid chain folds and bends based on the chemical features in the amino acid side chains. Through this folding and bending, the protein acquires its 3-dimensional structure. The completed protein, such as a membrane protein or insulin or a digestive enzyme, may be used within the cell or packaged for delivery via the bloodstream to somewhere else in the body where it is needed. Following the completion of translation, the mRNA strand may remain in the cytoplasm to serve as the template for producing another molecule of the same protein. In bacteria, an mRNA may last from a few seconds to more than an hour. In mammals, an mRNA may last several days depending on how long it lasts. The same mRNA strand may be translated hundreds of times. Eventually, it is broken down by enzymes in the cytoplasm. And that brings us to our take-home message for section 6.7. In translation, the information from a gene that has been encoded in the nucleotide sequence of an mRNA is read, and ingredients present in the cell cytoplasm are used to produce a protein. Moving on, why do some people get sick more often than others do? Why is one person taller than another? Which genes a person has certainly plays a role, but carrying a gene is not the only thing that matters when it comes to gene expression. Production of the protein that the gene sequence codes for, a person's traits, also depend on gene regulation, whether a gene is turned on so that it produces a protein product or is turned off so that there is no protein production. And this commonly hinges on a person's environment. Moving on, a microarray is a small chip that looks like a microscope slide and can be used to monitor the expression levels of thousands of genes simultaneously. The microarray is a powerful tool in the study of gene regulation. By taking cells from one part of the body, researchers can examine gene expression in that tissue. Microarrays are particularly useful in exploring how gene expressions differ in response to an illness or the treatment of an illness or in response to aging. For example, in a 5-year medical study exploring the health consequences of loneliness, microarray analysis of the immune system cells revealed that 209 genes were expressed differently in 2 groups of participants, those who were lonely and those who were not. Individuals in the lonely group had impaired transcription or decreased activity of some genes associated with an effective stress response. They also had increased activity in genes that are associated with inflammatory processes that are implicated in a variety of diseases. Related studies have shown a similarly reduced immune function among stressed out individuals. Moving on, cells have a wide variety of mechanisms that control when individual genes are expressed. One of the main ways of controlling expression is through proteins called transcription factors, which bind to specific sequences in the DNA called regulatory sites, often located in front before the promoter site of genes. The regulation may take the form of either positive control, in which binding of the regulatory protein initiates or speeds up gene expression, or negative control, in which the protein slows down or blocks gene expression. Regulatory proteins can be produced by the same cell whose DNA they are regulating, or they may come from nearby cells. During the initial development of an organism, for example, gene regulation is extremely important in directing cells to differentiate into one type of tissue, perhaps muscle, rather than bone. And proteins produced by one cell commonly influence gene expression in neighboring cells. Moving on, regulation may also be influenced by the nutrients an organism takes in. For example, the bacterium E. coli adjusts its diet depending on which sugars are available in its environment. Although E. coli prefers glucose, in the absence of glucose, it will use lactose as an energy source. The many E. coli cells that live in your intestine are likely to encounter lactose, which is the chief sugar in milk. The absorption and digestion of lactose, however, requires the expression of 3 genes, and bacteria do not devote energy to this process unless it is necessary. The regulation of gene expression uses a set of regulatory sequences called an operon, which includes a group of several genes and the elements that control their expression as a unit. The first regulatory site of the operon is the promoter. For a gene to be transcribed, RNAP must recognize and bind to the promoter region, the specific nucleotide sequence that signals the beginning of a gene. The second regulatory site is the operator. A molecule called a repressor protein can bind at this site and block RNAP from transcribing the genes necessary for lactose metabolism. The third regulatory element is a regulatory gene. This gene codes for the repressor protein that binds to the operator and blocks the RNAP. Moving on, here's how the operon, or what we call the lac operon, functions. If lactose is not present, 1, the regulatory gene produces the repressor protein. 2, the repressor protein binds to the operator region, and 3, the RNAP is blocked from transcribing the lactose metabolism genes. However, if lactose is present, firstly, the regulatory gene produces the repressor protein. 2, lactose binds the repressor protein, altering its shape so that it can no longer bind to the operator. And 3, with no repressor bound to the DNA, RNAP binds the promoter and transcribes the genes necessary for lactose metabolism. Note that the lac operon is switched on only in the absence of glucose. In the absence of glucose, a regulatory protein called an activator helps the DNA unwind in the vicinity of the lac operon, enabling RNAP to bind and transcribe genes. When glucose is present, however, the activator protein cannot bind to the DNA, and this prevents RNAP from binding to and transcribing the lactose metabolism genes. Well, genes can also be regulated in many other ways as well. And many eukaryotic genes, for example, have associated nucleotide sequences called enhancer sequences. When a regulatory protein binds to these enhancer sequences, the transcription rate for the associated gene is increased. This is a positive control. Additional mechanisms of gene control include the binding of chemicals to DNA that can block genes from being transcribed. This is a case of negative control, and the regulation of mRNA. mRNA can be regulated by altering its processing and transport, or by altering the rate at which it is broken down in the cytoplasm. Translation of mRNAs can be regulated too, as can the processing of proteins following translation. Increasingly, researchers are observing that the long-term activity of some genes can be influenced by alterations to chromosomes that do not change the DNA sequence. Moreover, these changes, called epigenetic changes in how genes are expressed, can be passed on to an organism's offspring. For example, in women who experience extreme stress when they are pregnant, one of the genes that influences how the hormone system responds to stress is altered within the fetus and remains so after the child is born. One mechanism by which this and other epigenetic changes can occur is that chemicals called methyl groups bind the DNA, resulting in a lifelong reduction in the rate at which the gene is transcribed. In this case, the silencing of this stress response gene is associated with a greater incidence of behavioral problems among affected children and adolescents. And that brings us to the take-home message for section 6.8. Environmental signals influence the turning on and off of genes. By binding to DNA, regulatory proteins can block or facilitate the binding of RNAP and the subsequent transcription of genes. Regulation of gene expression can also occur in a variety of other ways that may enhance or impede transcription, or alter mRNA's longevity and rate of degradation, or influence translation or protein processing. Moving on. Through the processes of transcription and translation, an organism converts the information held in its genes into the proteins necessary for life. But the process is only as good as the organism's underlying genetic information. Sometimes, something alters the sequence of bases in an organism's DNA. Such an alteration, called a mutation, can lead to changes in the structure and function of the proteins produced and have a range of effects. Sometimes mutations result in serious, even deadly problems. In other cases, mutations have little or no detrimental effect, and occasionally, but very rarely, they may even turn out to be beneficial to the organism. As an example of how mutations can affect an organism, consider breast cancer in humans. When two human genes called BRCA1 and BRCA2 are functioning properly, they reduce breast cancer risk by helping to repair DNA damage, which prevents the accumulation of changes that might lead to cancer. If the DNA sequence of either of these genes is altered through mutation and the gene's normal function is lost, the person carrying the allele has a significantly increased risk of developing breast cancer. Because a variety of factors, including environmental variables, are involved, it's impossible to know whether these individuals will develop breast cancer. Currently, more than 200 different mutations in the DNA sequences of the BRCA1 and BRCA2 genes have been detected, each of which results in an increased risk of developing breast cancer. Given the havoc that mutations can cause in an organism, it might be surprising that most mutations are neutral, that is, having neither a positive nor a negative effect on an organism. This happens when a mutation occurs in a non-coding region of DNA, or when a change in DNA within the gene doesn't alter the function of the protein produced. Researchers estimate that the rate of mutations in cells involved in reproduction is approximately 10 to the negative 8th base pair per generation. Mutations play an important role in evolutionary processes, from those heritable mutations that reduce an organism's ability to survive and reproduce to those that have no impact on an organism's survival and reproduction, to those extremely rare mutations that happen to be beneficial. In fact, every genetic feature in every organism was, initially, the result of a mutation. Most mutations you inherit from your parents will have no effect, and all of us are carrying mutations that we will never know about. Moving on, the type of cell in which a mutation occurs is important. Mutations in non-sex cells, such as skin or lung cells, can have bad health consequences for the person carrying them. Many forms of cancer, such as lung cancer and skin cancer, result from such mutations. Importantly, non-sex cell mutations are not passed onto your children. However, mutations in the sex cells or the gametes do not have any adverse health effects on those carrying them, but these mutations can be passed on to offspring. Individuals inheriting mutations from a parent can be at risk for diseases such as breast cancer or cardiovascular disease. Inherited mutations can also have an effect before birth, sometimes causing miscarriages or birth defects. The changes to DNA caused by mutations are generally of two types: point mutations and chromosomal aberrations. Moving on, point mutations occur when one base pair is substituted for another or when a base pair is inserted or deleted. Insertions and deletions can be much more harmful than substitutions because the amino acid sequence of a protein is certain to be affected. If a single base is added or removed, the 3 base groupings in an mRNA get thrown off, and the entire sequence of amino acids downstream from that point will be wrong. The reading frame is shifted. It's like putting your hands on a computer keyboard, but offset by one key to the left or to the right, and then typing what should be a normal sentence. It comes out as gibberish. Moving on, chromosomal aberrations are changes to the overall organization of genes on a chromosome. Chromosomal aberrations are like the manipulations of large chunks of tests, such as cut and paste, when editing a term paper. These aberrations can involve the deletion of an entire section of DNA or the relocation of a gene from one part of the chromosome to elsewhere on the same chromosome or to a different chromosome in the genome. They can also involve the duplication of a gene with a new copy inserted elsewhere in the genome. Whatever the type of aberration, a gene's expression, the protein that its sequence codes for, can be altered, as well as the expression of the genes around it. Moving on, there are 3 chief causes of mutation. Although one of them is beyond our control, the risk of occurrence of the other 2 can be significantly reduced. First is the spontaneous mutations. Some mutations arise by accident as DNA is duplicating itself. At the rate of more than 1,000 bases a minute in humans, when cells are dividing, DNA repair enzymes correct most errors, but not all. Second is radiation-induced mutations. Ionizing radiation, such as X-rays, has enough energy to disrupt the atomic structure, even break apart chromosomes by removing tightly bound electrons. Because ionizing rays cannot pass through lead, the lead apron you wear during a dental X-ray protects your body from them. However, even non-ionizing low-energy radiation, which does not remove electrons, can damage DNA. Ultraviolet rays from the sun, for example, can be absorbed by bases in the DNA, causing them to rearrange bonds. This can prevent them from pairing correctly with the complementary DNA strand and can transform a normal cell into a cancer cell. This is why long-term exposure to the sun can contribute to the development of skin cancer. Another source of dangerous ionizing radiation is found in nuclear power plants, where radioactive atoms are used in energy-generating reactions. The high energy of the radioactivity can pass through the body and disrupt its DNA, causing point mutations and chromosomal aberrations. Thirdly, we have chemical-induced mutations. Chemicals such as those found in cigarette smoke, vaping emissions, and engine exhaust can also react with the atoms and DNA molecules and induce mutations. And that brings us to our take-home message for section 6.9. Mutations are alterations in a single base pair or changes in large segments of DNA that include several genes or more. Mutations may be harmful, neutral, or beneficial. Mutations play an important role in evolution. Moving on, Errors in the genetic code within a gene are responsible for many diseases. In Tay-Sachs disease, for example, an individual inherits genes with a mutation that causes an inability to produce a critical lipid-digesting enzyme in the lysosomes, which function as cellular garbage disposals. Because these diseased organelles cannot digest certain lipids, the lipids accumulate undigested. The lysosomes swell until they eventually choke the cell to death. This occurs in numerous cells in the first few years of life and ultimately leads to the child's death. Although the details differ from case to case, the overall picture is the same for many, if not most, inherited diseases. The pathway from mutation to illness includes just 4 short steps. 1. A mutated gene codes for a non-functioning protein, commonly an enzyme. 2. The non-functioning enzyme can't catalyze a reaction as it normally would, bringing the reaction to a halt. 3. The molecule on which the enzyme would have acted accumulates, just as half-made products would pile up on a blocked assembly line. And 4. The accumulating chemical causes sickness and/or death. The fact that many genetic diseases involve illnesses brought about by faulty enzymes suggests strategies for treatment. These include administering medications containing the normal functioning version of an enzyme. For instance, lactose intolerant individuals can consume the enzyme lactase, which gives them the ability to digest lactose. Alternatively, lactose intolerant individuals can reduce their consumption of lactose-containing foods to keep the chemical from accumulating, and this reduces the problems that come from lactose overabundance. And that brings us to our take-home message for section 6.11. Many genetic diseases result from mutations that cause a gene to produce a non-functioning enzyme which blocks the functioning of a metabolic pathway.